Hello and welcome to an HDRN Pragmatic Trials Training Program educational module. I'm Liam.
And I'm Angela. Today, we're discussing a more technical topic about estimands, estimators, and analysis strategies in pragmatic clinical trials. We'll cover four main themes: the critical role of the trial statistician, the difference between estimands and estimators, common analysis strategies such as intention to treat, per protocol, and as treated, and how to select appropriate statistical methods for different outcomes.
To set the scene, pragmatic trials study how interventions work in real-world settings. Unlike explanatory trials that run under ideal conditions, pragmatic trials embrace the complexity of clinical practice. They deal with messy data because participants may skip visits, switch treatments, or add other therapies, which can dilute or amplify treatment effects.
To kick off, let's consider how not to involve a statistician, the hidden stars of research.
I have a study, and I need to know how many patients I need. I think I only need 3 patients.
Okay, what kind of study is it?
I'm doing a lab study. Can I just use 3 patients?
That depends. What are you trying to show?
We always use 3 patients.
Okay, what are you trying to show?
I'm trying to show that drug A and drug B are better than only drug A.
How are you measuring if it is better?
We are looking at apoptosis. Can we just use 3 patients?
What kind of measure of apoptosis? Do you know how variable that measure is?
We always use 3 patients. That is what we have published before.
3 patients per group or in total?
Yes.
So yeah, don't be the researcher who calls the statistician after the data are in and says, "I've got 100 patients. Can you, find a method that gives me 80% power?" Your trials methods must be designed, not retrofitted. The trial statistician must be a full team member who shares responsibility for design, sample sizes, randomization, data collection, and handling missing information, monitoring, handling stopping rules, analysis, reporting, and interpretation.
Having a statistician on board from start to finish is just good science. Okay, let's clarify the central concept behind the trial estimand.
The International Council for Harmonization Guideline on Statistical Principles for Clinical Trials includes the addendum on estimands and sensitivity analysis. This report defines the estimand as a systematic description of the effect we want to quantify. The estimand tells us precisely what treatment effect we're aiming to estimate.
The estimand has several attributes. First, the treatment, which is the specific regimen being evaluated. Second, is the population, which is the eligibility criteria and target patient group. Third, the endpoint or the outcome measure of interest. Fourth is the summary measure, and how we summarize the effect across the population. The addendum also recommends specifying how to handle intercurrent events, like treatment discontinuation or switching treatments. In pragmatic trials, these events are common because participants may not adhere fully or may use other therapies alongside the study intervention. It's essential that your estimand specifies how these events will be handled, so let's walk through the main approaches.
Treatment policy means we analyze everyone in the group to which they were assigned, and we keep all outcome data even if participants stop or switch to another treatment. Hypothetical asks what results would look like if the event hadn't happened, and we model that scenario. While on treatment uses data only while participants stay on the assigned treatment, then we stop using data at the event. If that censoring might be related to prognosis, we plan methods to address it. When another event precludes the outcome, like death before readmission, that's a competing risk, and we use competing risk methods rather than ordinary censoring. A composite rolls the intercurrent event into the endpoint when it's clinically meaningful to count it as part of failure or success. Finally, principal stratum targets the effect in the subgroup who would show the same event behavior under either arm. That's useful, but it relies on extra assumptions, so we state them up front. always pre-specify which strategy is primary and list sensitivity analyses in your statistical analysis plan. If you change the strategy after seeing the data, you risk introducing bias. More on sensitivity analyses later. Once you've defined the estimand, the estimator comes into play. It's the statistical procedure used to calculate the numerical estimate of that effect. In other words, the estimand is the what and the estimator is the how.
Think of it like this. If the estimand is your destination, the estimator is the route you take to arrive at that destination. Each estimand requires an estimator suited to the outcome type and study design. For continuous outcomes, such as changes in blood pressure or body weight, linear regression provides an estimate of the mean treatment difference, often adjusting for baseline or other prognostic variables. For binary outcomes, such as whether a participant achieves at least 5% weight loss or experiences an adverse event, logistic regression is typically used. It delivers an odds ratio summarizing how the odds of the outcome differ between treatment groups.
For time-to-event outcomes like time to rehospitalization, we can use the Cox proportional hazards model. It estimates hazard ratios and accommodates censored data.
When data are clustered, like patients within clinics, or when you have repeated measures, you need to account for correlated outcomes. Generalized estimating equations yield population averaged, or sometimes you'll hear marginal effects, which align with a participant average estimand, that is, estimating the average effect across all individuals rather than within specific clusters or subjects. Mixed effects models include random intercepts or slopes and yield a conditional or cluster-specific estimand. Both generalized estimating equations and mixed models are valid, but they answer different questions. Generalized estimating equations results are about the average person, while mixed models tell you about differences between clusters.
A trap is to fit a mixed model but report it as a population average effect. Again, mixed effects models yield conditional effects. To get population average interpretations, you need to marginalize or use other tools. Always specify in your statistical analysis plan whether your estimand is participant or cluster average.
Right, and once we've defined what kind of effect we're estimating, whether participant or cluster average, the next step is deciding how to analyze it. The intention to treat principle addresses the first key question. What is the effective treatment assignment? ITT analyses include participants according to their initial randomized assignment, regardless of compliance or protocol deviations. This approach mirrors clinical practice, where not everyone adheres perfectly.
ITT preserves the benefits of randomization and tends to provide conservative estimates. Importantly, ITT estimates the effect of assignment, not the effect of actually receiving treatment. When adherence patterns differ across trials, the ITT effect can vary even if the biological effect is the same.
The ITT approach answers the question, if we recommend this treatment to all eligible patients, what will happen on average? But clinicians and patients may want to know more. What if I actually take the medication? That leads us to other strategies.
The per-protocol analysis addresses a second key question. What's the treatment effect among participants who adhered to their assigned protocol? It includes only participants who meet the inclusion criteria, followed the protocol, and had outcome data collected during follow-up. In effect, you exclude or censor data after non-adherence.
Per protocol analyses are useful for questions like non-inferiority, but they break randomization and require adjustment for potential confounders. Methods like inverse probability weighting on propensity scores can be used. If adherence patterns matter for safety outcomes, per protocol analyses can be informative, but they should be pre-specified and handled carefully. It's also worth noting that per protocol analyses often show larger treatment effects than ITT, because you're looking at the subset of participants who actually received the treatment. But those larger effects could reflect who adheres, rather than the true treatment effect. That's why transparency and confounder adjustment are key.
The third key question: what is the effect of the treatment that participants actually took? This question can be answered using an as-treated approach, where participants are reclassified according to the treatment they actually received, regardless of randomization. They can shed light on the undiluted effect of the treatment, but ignore randomization and are highly susceptible to confounding. As mentioned earlier, to mitigate confounding, we can borrow methods from observational research, such as inverse probability weighting on propensity scores. Even with these adjustments, as-treated results should be interpreted cautiously and reported as secondary.
Because the ITT, per protocol, and as-treated strategies each answer different questions, they correspond to different estimands. The key is to align your analytic strategy with your trial's objective so your primary estimand reflects the main question you want to answer. In pragmatic trials, the ITT estimand is usually primary because it captures the real-world effect of assigning treatment. That is, what would happen if this policy were implemented broadly. The per-protocol and as-treated analyses are often pre-specified as secondary, helping to explore the effect among participants who actually adhered to treatment. That said, in non-inferiority or equivalence trials, per-protocol and sometimes as-treated analyses carry greater weight, since poor adherence could bias results toward falsely concluding treatments are similar.
Yeah, so building on that idea of matching the analysis to the estimand, the same logic applies to cluster randomized trials. In these trials, your estimand might target either the average individual across clusters or the average cluster itself. Again, a participant average estimand reflects the effect for an average person across all clusters and is usually estimated with marginal models like generalized estimating equations. A cluster average estimand, on the other hand, reflects the average effect across clusters and is often estimated with cluster summaries or mixed models.
A brief methodological reminder to start. Conventional regression assumes independence among observations. In clustered data, such as patients within hospitals or residents within long-term care homes, the assumption of independence is violated. Ignoring within-cluster correlation typically yields underestimated standard errors, inflated type 1 error rates, and confidence intervals that are too narrow. Cluster-aware methods are therefore required.
For trials targeting a participant average or marginal estimand, generalized estimating equations are appropriate. However, when the number of clusters is limited, apply small sample adjustments to the robust variance to reduce bias in standard errors and improve type 1 error control. Some comment sample size corrections include the Kauerman-Carroll, Fay-Graubard, or Mankel-de Rouen.
If the analysis uses mixed effects models with random intercepts and/or slopes, remember that these models yield conditional or cluster-specific effects. With only a few clusters, use Kenward-Roger or Satterthwaite degrees of freedom adjustments for fixed effects models. These corrections account for uncertainty in estimated variance components and provide more reliable inferences.
When model assumptions are difficult to defend, consider randomization-based checks as a sensitivity analysis. Permutation tests that respect the original cluster randomization can provide exact or near-exact p-values under the sharp null and help assess whether model-based conclusions are robust.
Another practical issue is informative cluster size. That is, situations in which larger clusters differ systematically from smaller ones. Standard marginal estimators can then be biased. Methods that give equal weight to clusters, such as independence estimating equations, IEE, or direct cluster-level summaries followed by appropriate inference, can mitigate this problem. Pre-specify the approach and rationale in your statistical analysis plan.
Finally, guard against model and target mismatch. If your primary estimand is participant average, either use a marginal method or marginalize mixed model estimates before reporting. If you intend to use a cluster average estimand, say so explicitly and use methods aligned with that target. A clear linkage between the estimand and the estimator should be specified in the statistical analysis plan and reiterated in reporting. Here, we present a hierarchy of sensitivity analyses while keeping the estimand central. We begin with the primary analysis, which must be pre-specified in the statistical analysis plan. For superiority trials, this is typically intention to treat, which estimates the effect of assignment. For non-inferiority or equivalence trials, it is good practice to pre-specify both ITT and per-protocol analyses. Non-inferiority should be demonstrated in both approaches. The next item is per-protocol. Define adherence criteria a priori, such as exposure windows, allowable deviations, censoring rules, and so on. Because conditioning on adherence breaks randomization, plan confounding adjustment when targeting the per-protocol estimand. Then, as treated. Participants are analyzed according to the treatment actually received, which is often time-varying. Valid inference again requires adjustment for time-varying confounding. As treated is rarely primary, but is informative for adherence and implementation questions. Missing data analyses assess sensitivity to unverifiable assumptions. Compare missing at random and missing not at random using multiple imputation, inverse probability weighting, and simple tipping point analyses to show how conclusions change under alternative mechanisms. Finally, model robustness keeps the estimand fixed while stress testing the estimator. Vary link functions, working correlation structures for cluster data, and apply small sample corrections when there is a small number of participating clusters.
The rationale for these analyses is interpretability. Each item addresses a distinct question, so misalignment of estimand, estimator, and analysis can mislead. For instance, a hypothetical strategy that estimates outcomes as if discontinuation had not occurred answers a different question than the ITT approach, where we use outcomes from all participants regardless of their adherence. Both may be appropriate, but they inform different decisions. To avoid misapplication, explicitly state the estimand and map the primary and appropriate sensitivity analyses in the statistical analysis plan.
Let's summarize the main takeaways. 1. Engage a statistician early. Their expertise spans design, data collection, monitoring, analysis, and reporting. Pragmatic trials involve complex issues like adherence and clustering, so statistical expertise is essential. 2. Define your estimand clearly. Specify the treatment, population, endpoint, summary, and how to handle intercurrent events. Align the estimand with your clinical question and stakeholders. 3. Choose an appropriate analysis strategy. ITT analyses estimate the effect of assignment and maintain randomization. Per-protocol analyses estimate the effect among adherent participants but require adjustment for confounders. As treated analyses reclassify participants by treatment actually received and are prone to confounding. 4. Select the right estimator. For example, use linear regression for continuous outcomes, logistic regression for binary outcomes, Cox models for time-to-event data, and generalized estimating equations or mixed effects models for clustered data. Finally, pre-specify your analyses and report transparently. That is, clearly state and report all of the above-mentioned items in the protocol and statistical analysis plan.
Well, folks, that's a wrap. We hope this module helped demystify estimands, estimators, and pragmatic trial analysis. Thanks for tuning in.